Hi, my name is Cameron. I'm in my third year of biotechnology at Queen's University. I'm on the human practices team on Queen's QGEM, and today I'm going to be explaining synthetic biology at 5 increasing levels of complexity.
So Jackson, thank you for coming today. Before this, we actually had a little conversation about strawberry DNA.
Yeah.
So based on our discussion, what do you think DNA is?
DNA, what I think is it's a bunch of little things making one big thing.
That's exactly what it is. I like to use the word big here because there's so much DNA in your cells. There's about 2 meters in each cell. If you take all the cells in your body, that's trillions of cells, and you stretch out the DNA, it can go to the moon and back 150,000 times.
That is.
And in the same distance, actually, it can even go around the whole solar system 40 times. So there's a lot of information in your cells.
Absolutely.
And like you said, it is a small thing that makes up a bigger thing. So what DNA is, it's essentially the code of life. In that code are all the components that make up a cell. So DNA is made up of 4 different letters.
Okay.
And those letters, just like the alphabet, are put together in a language, just like letters make words. And those words then code for things called proteins. And you know what a protein is?
Yeah.
We had some proteins earlier. We went out and got some eggs. There's some protein in eggs. And those proteins carry out the functions in your body. And they create all the components and carry out all the chemical reactions. There are things like enzymes. And enzymes are responsible for speeding up those reactions.
Yeah.
And different DNA in different organisms are a really important way that we distinguish between those organisms.
Yeah.
Like, I also remember one time you're telling me the difference between a tortoise and a turtle.
Yeah.
What makes a tortoise different from a turtle is the code in their DNA.
Yeah.
Yeah. So, one cool thing we can do with DNA is we know how to read it.
Yeah.
And if we know how to read it, we can write it. And so what we can do there is we can take that strawberry DNA that we were talking about, and you can actually introduce that into different things. And that organism that takes it up, let's say we put that into a bacteria. That bacteria can then start producing that code of the strawberry.
Yeah.
Yeah. So that's one side of it. And today we're talking a little bit about synthetic biology.
Yes.
And so when you hear the word synthetic biology, what do you think of?
Well, I think of biology, like living things, like animals and plants, and I don't really know about synthetic.
Yeah, so when we use the word synthetic a lot of times, it has a bad connotation a lot of times. It means kind of human-made.
Yeah.
But really what synthetic biology is we're taking life and we're applying an engineering principle to it.
Wow.
So if I asked you then, what's the difference between a scientist and an engineer? Like, what would you say?
Well, scientists are more like researchers, and they learn about there's different types of scientists,
Yeah.
that's I know. And engineers are more of like, they build stuff, they help our planet like revolve, and yeah.
Yeah, that's exactly it. Scientists are responsible for investigating the natural world around us, understanding why certain things happen the way they do. And then engineer take those principles that we figured out, and then they apply them in that way. And that's what synthetic biologists are. They take the information that scientists discover, so that DNA codes for information, codes for proteins. They take that, and then they try to engineer DNA to solve different problems. So scientists and engineers have used this to solve problems in agriculture and produce medicines from bacteria and scale this up. So the key difference between a scientist and an engineer and a synthetic biologist is that a synthetic biologist is a type of engineer that takes life, engineers it to solve problems. And that's, you can do pretty much anything as long as you fall within the rules of life, really.
Yeah.
So some of my favorite examples are using microbes to survive in space, to engineer plants to produce more crop yield, to using bacteria to solve medicines. So if you could be a synthetic biologist, so you are a synthetic biologist, what kind of problem would you want to solve? What would you want to do? You can be for anything. It could be, I want to make a bacteria glow. Or
Probably like world hunger. I
World
know that's
hunger.
a pretty big problem, and it may like never be solved. But if we do find something that can solve it, I think that would be great for our world.
Wow. Yeah. That's a huge goal right now for a lot of work in synthetic biology. Even here in Kingston, we have a company called Performance Plants. They're right now working on looking at the genes of plants and seeing which ones can be incorporated into the plant to produce crop yield or be more resistant to altering temperatures. But the thing I think I find most interesting about this is we aren't the first people, like we aren't the first things to do genetic engineering and work with synthetic biology.
Yeah.
Do you know which organisms did that first? What do you think? Take a guess.
Apes.
Apes? So I'm even going back further than that, but it's true that we're really similar to apes in terms of our DNA.
Absolutely.
I think the number is around like 97%.
Yeah.
But the first real genetic engineers were actually bacteria and viruses, and that goes back billions of years. And we actually took those tools from bacteria and viruses, and we use that just in our own applications. So we haven't really invented anything. I think we more took what was already there and then applied it to solve some issues today.
Yeah.
And that's pretty much what genetic engineering is in synthetic biology.
It's amazing. Yeah.
Yeah. So what do you think? Have you learned anything?
I really learned what synthetic engineering. Well, yeah, and more about like scientists and genes, biology, stuff like that. It's
Yeah.
really nice.
It's so cool what you can do with just the information that we have available to us.
Yeah.
And use it to solve problems.
Absolutely.
Yeah. So thank you.
Thank you.
How are you go? Thank you for coming in today. So what I understand is you're in grade 12, right?
Yep.
But you're not taking biology right now.
No, I'm not. I took physics and chemistry, and so I haven't even taken grade 11 biology. So
Okay.
it's a new field.
Yeah, yeah, exactly. But a good thing about biology is to understand biology, it's really fundamental to understand physics and chemistry. So you already have that background. So we're going to be talking a little bit about DNA, genetic engineering, synthetic biology. So what do you know about DNA? What have you heard about DNA?
I mean, I guess I have a really superficial idea of DNA. It's obviously what the makeup of life. Friends he programs, and we kind of chat back and forth about what programming actually is. And I like to see DNA as the sort of the characters that make up what things actually do or are, in a sense.
Well, it sounds like you know exactly what DNA is, because that's exactly right. It's a molecule, deoxyribonucleic acid, and it's a code. There's information. The information in DNA is primarily used with 4 different letters, 4 different bases. Just like the languages, you have letters that make up words, you have DNA. You have these 4 different letters that make up words, except in biology, they're called codons. And those codons, those 3 bases, then code for a specific amino acid. That amino acid makes up proteins. You know what protein is?
Yeah.
So, and then those proteins are then responsible for going out and producing the characteristics that you see in a cell. It's what makes a plant a plant. This isn't a real plant, but it's what that does and what makes a human. And we actually share a lot of that information. We're half of a banana. It's a universal language that we use, and we can actually read and understand. So what happens at the fundamental level is you have DNA, you have those different codons, and that is transcribed into something called RNA. RNA is essentially a transient form of DNA. It kind of snapshots the information, but we don't want to mess with our DNA because it's really important, and that's where you have cancers, when there's mutations in your DNA. So what happens is it's transcribed into RNA. That RNA can then go and transport outside of the nucleus where the DNA is stored, and it's translated into proteins. And that's when you have, like, proteins are the machines of the cell, and they carry out chemical reactions, and that's kind of how the cell works. What's interesting about genetic engineering is we actually have technologies. Well, we don't really have technologies. We kind of took our technologies from bacteria and viruses and used them to edit that code. A lot of people suffer from genetic dispositions. Have you heard of genetic diseases?
Yeah, I'm sorry, when you do that, are you,
Yeah.
I might be both, but are you putting in new compounds to make different proteins, or are you taking out stuff, or what is that?
When you're doing genetic engineering?
I guess so. Yeah.
So, well, what they do, how it was traditionally done, is you actually put in the genetic information that you wanted and then into the cell. Now what we can do is we can actually go in, fix the word, and make it produce the compound we want. So you can introduce information, and the machinery in the cell can read the information you give it, the DNA you give it.
What is the process of, I guess, fixing the word looks like? Are you...
Yeah, so it's actually really cool. How this was discovered was actually in a yogurt factory. And bacteria that are in yogurt, there's a lot of good bacteria in yogurt. They were infected by viruses. And to combat viruses infecting these bacteria, they had these proteins. These proteins in the bacteria recognized when the virus infected and inserted the viral DNA. So viruses, how they attack bacteria is they insert their own DNA. That DNA then integrates itself into the DNA of the bacteria, and it kind of hijacks the whole cell. It overtakes its machinery and starts producing more virus, and is really, really bad for the bacteria, because now it essentially is hijacked, it pops and releases more virus. So the bacteria has an enzyme that can recognize the DNA of the virus and cut that DNA. So there was the Nobel Prize that was won of taking that enzyme, so that protein, inserting a target sequence that it knows where to cut. So that's how we do it.
So an enzyme and a protein is they're sort of the same thing.
So protein. Not all proteins are enzymes, but most enzymes are proteins. So enzymes are just proteins that speed up the reaction rate. So in chemistry, when you're learning about catalysts, it's basically it's a biological catalyst.
Okay.
So it speeds up the rate at which a molecule is transported. So if you use an enzyme or converting one molecule into another, an enzyme will bring them together and speed up those reactions. So that's it's just a protein that carries out a specific function. That's
one question. I guess when I think of genetic engineering, I guess I like to picture the DNA sequence, and I like to picture maybe like tweezers coming in and like taking something out, putting something in, kind of replacing things. Is the enzyme the I guess the tweezer in the analogy, is that the thing putting in and taking out stuff?
Yeah, that's exactly it. And so the enzyme does that. It cuts it at a specific site that you want to cut it at. And originally what we had to do was we had to cut the DNA and then insert some DNA in between that. Now what we can do, and it's actually, they just used a gene therapy on a newborn child a few days ago to cure a genetic disease. And what they do now is they actually made those enzymes even better. So those proteins, they made them even better at targeting. And now what they can do is they go in, take that letter, so the code, and they can change the letter. Instead of cutting now, they can just change it.
So you go from like physically like going in and deleting to like, how do you, I guess my question, how do you program an enzyme to accomplish a task?
That's a great question, and that's what they won. That's what Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize for figuring out. So typically, when you have a protein in a bacteria cell, it needs to recognize and cut the viral DNA. But if it's recognizing DNA and cutting DNA, it's going to cut its own DNA. So we actually took the mechanism from bacteria, where there's something called a PAM site. And a PAM site is just a feature of the code. So it's just another code that the enzyme can see recognize, and then cut. So you can actually manipulate that site. So you can choose where you want it. You can choose where you want the enzyme to target by looking at that recognition sequence.
And I guess you can sort of train it to like detect a certain strand of DNA and kind of rewrite it. Is that, yeah?
Yeah, absolutely. And we're getting better at it too. There's even new technologies that use different types of recognition that are even more specific. And it's we're really just taking what was already in nature and trying to improve it a little bit not even improve, but use it for a different purpose. But now they're getting really, really good at using these proteins and enzymes to be really, really specific, to change even just one of the code. So one of the bases is what we say. So one of those letters. And that's huge.
So one of the letters in the enzyme?
One of the letters in the DNA sequence.
In the DNA sequence.
Yeah, so there's those 4 letters.
Okay, so it's, what's the difference been between how we used to, I guess, I don't want to say make, but like use enzymes, say like 50 years ago to now? Like because like you mentioned, there's been huge advancements.
Yeah.
And there's like a obvious gap from where we once were to where we are now. So what's the main thing that's changed? I guess basically, like if you had to describe what it looks like in a sense, how would you?
It's really, there's not one thing that changed the field, but it's actually a progression in technology as well. So originally back in the 70s, what they had to do was they used different enzymes, not the ones we were talking about earlier that cut specifically, but they used things called restriction enzymes that cut less specifically, but they basically cut the DNA and then you can insert a new fragment in. So that's a little bit different from recognizing a specific sequence and then changing it like that.
Is it different enzymes that cut on different enzymes that insert, or is it the same one? Or?
So those enzymes, those different enzymes are called restriction enzymes, and those are also found in bacteria. So they're less adaptable to certain sequences. They recognize certain sites, except you can't really tell it which sites to recognize. It's an inherent property of that one enzyme. So we went from there using that, and it was pretty good. We even used it to produce medicines like insulin. We used that type of technology to revolutionize that field. But now with the onset of computer technologies and AI, it's really expedited the process of searching through DNA. Now you can sequence a genome. So you can go take a bacteria, read the entire genetic sequence, the entire language, I suppose, of that organism, and download it to a computer. And then you can search through the entire code to try to figure out why that organism is a certain way. So using that, you can then use that as a target for your protein that specifically edits different things. So like AI has been huge, and sequencing technologies has been big too.
I guess that's how you find patterns too, right?
Yeah,
you're able to like connecting it back to things like math. You're able to, in a sense, graph the information to a point where you can actually make something of it.
Yeah, I'm so glad you talked about it, because that's a huge intersection, really.
I imagine
now it's becoming even more like computer science, and you use these things. You use a lot of math, really, to try to go through and figure out what is happening in this. Like, there's a lot of information, and one of the big problems now is we actually have too much information. Like, we don't know what it all means, and we have to go through, use principles even principles from physics and chemistry and math to understand what we're looking at. And it's all integrated with each other. So yeah.
Now that you mentioned that about informing the public, what would you say some of the biggest misconceptions? Like, say you go to a party, you go to meet someone, and you're like, oh, actually, I'm in genetic engineering. What's a question they ask you?
Yeah, I think that one of the greatest misconceptions I think that I like to talk about is that it's not new. Genetic engineering has been happening since agriculture started. Like corn doesn't exist on its own. That's actually taking agricultural practices and breeding different crops together to produce desired traits, that is genetic engineering. It's just a slower form of it.
Just the
Exactly. So
I
we
imagine
want
like breeding dogs, for example. Too.
that's
like,
exactly
it's
right.
Exact same thing.
Yeah, it's just natural evolutionary principles. So like dogs, exactly. That's something called artificial selection. Dogs today are domesticated. We've selected for the traits that are most desirable, and that means, and so it's easy to understand that from a physical side. You can see the evolution of like the primitive dogs to our domesticated dogs that we have today. You can see that transition, but really what you're selecting for is their DNA. And that's what makes the difference between a domesticated dog versus a more wild, I don't want to say primitive, but previous ancestor.
Or
of
even
them.
like a sled dog that's been bred to do a
Absolutely.
task versus say like a Chihuahua where you could not pull a sled or anything.
Yeah, exactly. And so I think that's what I find is the greatest misconception. It's not like a very fresh new technology. We can just do it in different ways now. We can do it faster. And we are not the first organisms to do it, and we've been doing it for a while. So yeah, that's my favorite thing I think to talk about.
Sorry, one more thing I think of when I think of genetic engineering is Jurassic Park. Like and especially it was I don't know if it was a month ago or so that they were able to reintroduce this species that was previously extinct. What do you think of that? Or what do you think?
Yeah, so are you talking about the colossal biosciences bringing back the dire wolves?
Yeah, exactly. Yeah,
so right now it's a big discussion. There's a lot of scientists that agree with what they're doing and a lot that don't. Right now, I just want to start off that the technology that's used right now, it's impossible to bring back the dinosaurs because
science fiction.
Yeah, well, at least right now, because what they're doing is they're taking preserved DNA and they're reintroducing specific traits and genes and trying to figure out what makes a dire wolf and what makes a normal wolf a wolf. So what they did actually is they took the DNA from a wolf and they tried to introduce traits that they've identified as coming from the dire wolf, but they haven't actually resurrected the whole organism.
That's really interesting.
They've kind of tried to see which genes, so which stretches of the DNA and which codes for a dire wolf, and
so
then
it's sort of like giving a car paint job. It's like you have a base and you're like well dire wolves are this way and this way and this way, and say strands A, B, and C make up these things about dire wolf. We can just kind of throw them in. We can paint it over, and then we have a new car. Say it.
Yeah, and another example of that came from the same company is they introduced the woolly mammoth gene for the fur. They introduced that into a mouse, and you produced a mouse with woolly mammoth fur. But is it a woolly mammoth?
No.
Like, not exactly. But
No,
they've done a really good job on it, making it look like it is. But then again, what their main goal there is to reintroduce lost genetic diversity, which genetic diversity is so having different organisms with different genes and traits is really healthy for an environment.
Yeah,
and
that's what you need for a healthy and a sustainable environment too.
Exactly. Yeah.
No,
I think that's everything. Have you? What do you think? Have you learned something today?
Definitely. I mean, I really know nothing of biology. I think it makes more sense to it now, and to me at least, the way you described that, I can definitely think of the process, and I can definitely think and picture what the technology actually means and what it's doing. But you kind of left me wanting more. I want to I'm actually really glad because it's something I want to go home and look up, because it's definitely a really cool field, and it's got so many possibilities.
Yeah, absolutely.
Especially for you guys who are working with it every single day. I imagine it must be super exciting to just know that there's so much you can do.
Absolutely. It's an exciting place to be, and the future is equally exciting. So
yeah.
Thanks so much for coming.
Thank you. It's great.
Hi, Claire. Thanks for coming.
Thank you for having me. I'm so excited.
So last year, you were on the iGEM team.
I was, yes.
Yeah. So would you be able to tell me a little bit about your project last year?
Of course. So our 2024 iGEM project for QGEM was very exciting. It was for Parkinson's disease, and we were in the therapeutics village. So what we were designing was kind of like medicine, kind of something for Parkinson's. That's where it started. And then throughout the journey, it developed into like a pharmaceutical type of like molecule. And I think the biggest part about it was the molecular switch. That was kind of our novelty and the whole synthetic engineering part of it. And for this, there's a lot to it, so bear with me. So there's lots of parts, lots of names. So it starts with Parkinson's disease, which is when proteins that are normally found in the body called alpha-synuclein, they start to misfold and they can like aggregate. And our project, based on Brack's hypothesis, was that these aggregates begin in the like enteric nervous system in the gut and travel via the vagus nerve to the central nervous system and cause all the symptoms of Parkinson's, like the muscle like problems, like fatigue, all of the kind of emotional aspects of it as well. So that was the gut-brain axis. That was part of it. That was one of the triumphs too. A lot of people, it's very split in the scientific community if this is even like a real hypothesis. So that was definitely a struggle of the project. But based on that hypothesis, we were targeting the aggregates in the enteric nervous system. So the neuroendocrine cells is where this therapeutic would target. And the molecular switch inside of a little spore, like a probiotic, it would be taken. We'd hopefully add it to, like it could be added to yogurts maybe one day, but we'll see how that goes. See if anyone wants to take it over in the pharma industry. But the alpha-synuclein aggregates, we created a switch that would bind to them. But what was really cool about it is that it had like an input domain and an output domain. So the input domain would bind, and that was cyclophilin D. So it would bind to the C terminus of a misaggregated, misfolded protein of alpha-synuclein. And then upon that binding, the switch would like conformationally change, and then this would release neurosin, which is KLK6. And KLK6 has the remarkable ability to like cleave these aggregates, but you want to cleave in the right place. So by cyclophilin D sitting on the C terminus, it like holds it there, so then the KLK6 cannot come bind there. It would be cutting on the NAC domain, which is what we want. So it would just cut up the little aggregates and they'd be excreted through the body. And hopefully it would help Parkinson's patients, hopefully early stage, because that's where it kind of manifests is in the gut. I know there's a lot, so if you have any questions about that, but it's really, really cool.
Yeah, that was so cool. Like, how did your project even start last year? And kind of, like, how was it designing this molecular switch? Like, you guys did some modeling work and some wet lab work. So, like, what was that like? And how hard was it, I guess, also?
Oh, how hard. It was definitely a very ambitious project. We met with many, many experts, and some of them were like, whoa, like, okay, do you think this is going to work? Or, like, they were a little bit, not negative, but they were being realistic, because it is a very ambitious project. We only have one summer to do it, which they gave really good advice still. Like, nonetheless, they were really supportive. So from the beginning, it was an idea from our director. It was, like, her idea to, like, do this, like, the alpha-synuclein, the, like, neurosin, cleaving it. So she kind of started that, and then our amazing, like, wet lab team, they did so much amazing research, found, like, cyclophilin D, which made the switch, and just so many, it was a cascade of a bunch of ideas. And yeah, it was a lot of work. And unfortunately, I wasn't in the wet lab as much. I was for outreach, which was amazing to connect with the community and patients. But I know wet lab, they did a lot of like modeling online to find the right like binding sites and where to actually place things on the protein and everything like that. But it was a very ambitious project, but it went really well. We had really good results at the end.
And with you being the outreach lead last year, you're kind of the perfect person to ask this. But how exactly, like, what's your take on what synthetic biology is, and how did you explain that to your team, to the community? Like, do you have a definition?
Okay, so I'm not a dictionary definition, but to me, like, synthetic biology just means creating synthetic, like something new, something that's never been done before. And what's beautiful about the whole community, iGEM, even outside of iGEM, because synthetic biology is like worldwide, it's everywhere, is that it like encompasses so many different areas of life. There's like technology, there's agriculture, and being at the Jamboree, you got to see synthetic biology in its whole. So there are projects from like fashion, using like one of the projects, I think it was U of A, like University of Alberta, they were using like this wool created by spiders to create like fashion and clothing and stuff. Like there were so many amazing things. So synthetic biology is just so wide, and it can solve so many problems that we face in our world, which is just why it's such an amazing thing and great to be a part of.
Yeah, I think synthetic biology, like the word, it's not exactly a technology. It's really is like a way of thinking, a community, and iGEM is the perfect embodiment of that.
Is,
You go to the Jamboree, you see what everyone else is coming up with, you connect with other teams, and really, it's kind of like a co-inspiration, like you inspire each other. So, and I think now with new technologies, it's really a perspective of looking at life and seeing really what problems you can tackle using engineering principles, like design, and I'm sure there's a lot of troubleshooting and
lots of troubleshooting
and improving, and that's something your team is definitely familiar with, and what we're all working this year, and you'll get more hands-on experience with that in the lab. So it's really good. And I remember even hearing about a project you're just talking about, silk. There was even one application I was listening to, I heard of, is using silk, like antimicrobial silk, so that like using synthetic biology to introduce these genes into E. coli and produce silk. And I mean that's a great application to bandages, and when you have
like
open wounds,
exactly.
And preventing infection. This is a huge. So I really think that it's a great community of creativity.
It is. It's a very creative industry, and it's amazing.
Well, thanks so much, Claire. We kind of already talked about your, I guess, favorite things at the Jamboree. Anything else that stood out, really, or?
Yeah, there were some really, really cool projects. I would say there was definitely one in particular that I enjoyed and that I actually saw out, because me and my co-lead, we went through so many of the websites. Like, we were stalking, seeing what people were up to, how their websites were looking, and I came across one of them when we were doing it. I'm like, I have to see them at the Jamboree. Like, I want to see how this goes and what comes of the project. So it was for NYU, and they were creating a sensor for iron deficiency anemia. So usually, I am iron deficient, so this is why it's close to home. Usually, I have to get my blood tested. If I want to see my iron levels, they take some blood, they test my levels and everything. But they were coming up with a salivary model where you just take a saliva sample and it would test your salivary ferritin levels and detect how much iron is in your at least saliva. So it's not completely accurate, but having a new diagnostic technique for that would be amazing, because then I could just be going about my day, and if I'm feeling really tired, I could be like, maybe my iron's really low. Just this quick saliva sample and be like, oh, it is really low. I should really take some iron supplements or figure that out. So it was really, really cool, and I met them at the Jamboree, and they told me all about their project, and it was really cool, and they had some really good results, definitely some promising results for that.
Wow, that's great. I think a big part of it also is synthetic biology can make technologies more accessible.
Yeah.
Because biology is so efficient. We're really not better than, I think, 4 billion years of evolution, and there's so much diversity of organisms that are specialized for certain things, and I think there's really a bright future for using biology to solve problems, and diagnostics is definitely a cool application.
I know, yeah.
Well, that's all. Thanks.
Thanks for having me.
Yeah.
Neil, thanks for coming today.
Thank you.
So, from what I understand, you're finishing up your PhD.
Right.
And what exactly are you looking at in your PhD? What are you studying?
Yeah, so I work in the Department of Chemistry, which makes what I do in the Department of Chemistry quite unique, in that we do probably equal parts biology and chemistry, and what we do at the interface of those two disciplines is look at natural product biosynthesis, and this is essentially molecules that are produced by living organisms that aren't necessary for their survival, but over the course of evolution, they've evolved to produce these molecules to give themselves an ecological or evolutionary advantage. And therein, I guess it follows that the only reason nature has kept producing these molecules and sort of donated energetic cost into continuing to produce these molecules is because there must be some advantage. That's why it keeps making them. And so a prime example of a natural product is penicillin, right? Penicillin was discovered on moldy bread, and then we found out that the mold is producing that because it's antimicrobial, and now it's one of probably the biggest life-changing natural product that's changed the world in the last century or so. So that just goes to show the impact of natural products research. So what I look at is the sort of the biosynthesis, how these organisms make cool molecules, and can we find any that are new and novel that we can turn into drugs.
Wow. So run me through kind of the process of how you start that. Do you have a product in mind, a target in mind, and then you go back through evolutionary history, or how exactly do you start, I guess, looking for natural products? Like penicillin was
kind of an accident, right?
Right.
Is that what? Do you start with an accident, or how does that work?
Yeah, so happy accidents are always welcomed, but you can't rely on them in this day and age. Unfortunately, you're not going to write a grant saying, "Oh, I hope serendipitously to run into the next big drug," right? What we work at mainly in the Ross lab in the Department of Chemistry, which is I am co-supervised by both the labs of Avina Ross and Dr. Graham Howe. Dr. Ross's lab focuses on marine natural products, specifically natural products produced by marine bacteria. And so we know marine bacteria are, it's interesting because you can sort of see or guess where a natural product came from if you know what you're looking for. Marine natural products tend to be brominated, so a lot of these natural products that we work on have these bromine groups. Bromines are very large halogen, and it's interesting that, what role does, bromination have in the context of producing a natural product, and what kind of associated activity is associated with bromination of a natural product. Well, it turns out the ecological environment there sort of uses bromine for signaling. Like a lot of corals will find these brominated natural products and be like, okay, we should shoot our eggs or shoot our reproductive stuff over there and settle our corals in these brominated areas. Probably because in those brominated areas, there's not a lot of bacteria to compete with, because these, brominated areas have natural products that are inherently antimicrobial. So we've known that, but again, we're not exactly looking for that. We just know that's a thing. And so now that we're in the age of terabytes of sequence data freely publicly available online, what we've developed are databases and tools that can find certain motifs. And we use what's called hidden Markov models, which is a popular and very prolific bioinformatic statistical tool to sort of find things that look like halogenases, which are enzymes that install halogens. Because we know if there are brominated natural products, there must be an enzyme that installs that bromine on that natural product, and it has to be from a halogenase. And halogenases have a very characteristic amino acid sequence, which we can find using an HMM, hidden Markov model, and that'll point us to, okay, maybe this gene and its context will give us some information on how these natural products are made. So basically, to answer your question, we sort of go online and just like we put our tiny chemist brains together, and hopefully we get one brain cell out of that and find something new. So
well, it sounds like there's a few more brain cells and maybe one on trying to undergo that task. But that's so interesting, and I love that you're talking about the sequence data now.
Yeah,
like when it's funny, we've talked about already kind of the difference between genetic engineering and synthetic biology. When the term genetic engineering was coined, like back in the 70s, that was really limited in terms of what we could do with software. Now, we kind of needed a new term with like synthetic biology, where we're incorporating now streamlined processes of data. Like life now, you can essentially download the information of an organism onto a computer, and then you run like your hidden Markov models to go through that data and try to understand it. And we're very much still in the era of trying to understand what the data does and how you can use that. So it's so interesting. And now you're talking about, now that I have you here, actually, I have a kind of an interest question that recently I've been interested in. I've been reading a book about synthetic biology, and in that, we learn about the handedness of molecules. And in organic chemistry, we learn about the handedness of molecules, how you're enantiomers. And since you work on enzymes are specific for how those molecules rotate plane polarized light. Have you heard at all anything about mirror life before?
No, but
okay.
I think I know where this is going. It's piqued my interest.
Yeah.
Please continue.
So I guess the proposition is, if you could create amino acids that weren't in the left conformation and they were in the right, you would then be able to create life that's resistant to viruses that have evolved to infect those cells. Everything interacts depending on the chirality of the molecule. So do you think that is possible, I guess? Like you're working with enzymes and they are very specific. What if we altered the chirality? Do you think, what's your opinion on that?
Yeah, that's fascinating. That is very fascinating. Yeah, so there's there are a lot of people who study the prehistoric origins of life. And one of the big questions is how we have homochirality, right? We have D sugars predominantly and L amino acids. And the question is why?
Yeah.
Why did we land on that? Well, there's also a bit of contention, and a part of my research looks at these two fighting forces between enzyme specificity and enzyme promiscuity, because in some sense, for molecules to evolve and to select for new phenotypes, you have to have a version of high throughput screening. You can't have specificity all the time, because then you become it's the equivalent of having a very shallow genetic pool. You can't have new phenotypes if you aren't able to adapt, and part of adaptation is having promiscuous enzymes that can make new molecules to select for. So there's that, and there's fighting against enzymes becoming more efficient at making the same product. So where we get homochirality and or production of molecules with the same handedness as an advantage. So yeah, that's interesting. And there are enzymes too that in natural product biosynthesis called epimerases, which their job is to change L amino acids into D amino acids.
Really?
So hypothetically, you could have an epimerase that is selective for ideally one that can epimerize every amino acid. That's not likely, but you can have an epimerase system that changes the L amino acids to D amino acids, and then maybe some kind of translation system that builds D polypeptides, and then you'd have a D protein that has completely different chemistry, or it does completely different chemistry than the L version. Or you could have enzyme replacement, where you replace enzymes that make or act on the L enantiomer, and only produce the D version of that molecule, the D version, and then have an organism that is resistant or has emergent biological properties that we don't see in our homochiral universe, right? So very interesting, and I don't know what kind of implications that has, but certainly resistance to certain pathogens and having a completely, I guess, mirror version of every biosynthetic pathway is really fascinating. And I wouldn't be shocked if there was somebody who's trying to make a mirror bacteria or a mirror self-assembling microenvironment already, just to see what's going to happen. But that's a bit scary too, because if it is pathogenic, whatever that is, then how do we kill it? Well, I guess the answer to that question is chemistry. I mean, I just gave my exit seminar, and I think I wanted to end it with a bang. And the idea was that chemistry is the study of matter, right? It makes up this table, it makes up this plant, it makes up you and me, right? And if chemistry is the study of stuff, and there's a problem with stuff, how do you fix it? With stuff, right? So you got to understand the stuff. So everything is chemistry, and every problem and every pathology can boil down to molecules and how they interact with each other. So so that's why I do take pride in my chemistry background, although I'm applying it very biologically.
I love the intersection of biology and chemistry, so I'm right there with you.
So like, if you had a piece of advice to give somebody who wanted to enter like a biotechnology program, what would it be? What would be with regards to I don't know, choosing or working on a set of skills, or choosing a set of courses maybe, or doing some kind of supplementary learning to succeed in in that field, whether it be grad school or industry? What advice would you give?
I would say follow your interests, but be open-minded. I think just because you don't learn something in school doesn't mean you don't have to learn it. It's a lot easier to learn in school. That's the whole point, to once again streamline that learning process. But there are also so many resources to learn things on your own. So I think it's also a lot of the times people being forced to learn something, it's a little more difficult. So coming into it, I think follow your interests, and you will learn how to do it. but also be open-minded about it. Some things I've sounded hard, like coding is notoriously sounding difficult, but then I took the class and I loved the class. I'm like, this is so fun. There's so much you can do. There's so much automation you can achieve from this, and there's always more you can learn about it. But I think, yeah, being open-minded and also following your interests, because no one likes to learn something they don't want to learn if they don't see an application for it themselves. But,
Right.
Yeah.
Yeah. And before we close out, I think just to piggyback off of that, I think there's a lot to say about having to learn something under the right context. I think if something doesn't pique your interest the first time, it doesn't mean it's not for you. For instance, like I have a great anecdote for this, because I took second year organic chemistry, and not that I hated it, but I didn't really it didn't strike me as something that I wanted to pursue, and that was okay with me, because I still had a lot of biology that I could cling to, right? And then by the time I was in third year, I just had the most amazing teacher for organic chemistry, and it changed my whole perspective on what chemistry means and how I could apply that to the biology that I love so much. And I don't think I would be here if it wasn't for that. So sometimes you need a little bit of luck, a little bit of tenacity, but I do agree with you. Yeah, follow your interest. When you find something you love, you got to hold on to it tightly and run with it. Yeah.
Well, thank you so much, Neil.
Yeah, thanks, Cam.
So, Dr. Alliam, thanks for coming today. I've already had the pleasure to learn a little bit from you on synthetic biology already in the course Biochem 320, Applications of Synthetic Biology. So thanks for coming today to share a little bit more about that and kind of refresh, I guess, my memory a little bit on the course. But I want to start, I've been meaning to ask you when did your interest in synthetic biology start? When you decided to start running that course, I guess.
Yeah, so running the course, that came probably a couple of years ago, once we had a number of teams who were coming in to wanting to join a club, wanting to get some wet lab research experience, interested in the idea of things like bioremediation or designing new ways to use biological things to treat infections, whatever, provide clean water sources. And what we found was that the students had a lot of interest and creativity, but none of the tools to do any of it. And so after a few runs of the IGEM team, where the team is getting started in May, and they haven't accomplished anything until the end of June, and the competition starts in September, we realized that we needed to have some type of driver's ed course for these students to be able to have more of the tools on hand when they start in May, to then be able to be a functional team much before they were.
Yeah, I mean, I know I can speak to that definitely. Now joining IGEM after taking the course, we've learned so much about designing plasmids and about even a lot more on the ethics side of things too, which is a big part of IGEM as well. So it's definitely helped me right now. So I also wanted to talk to about IGEM and it really being kind of a community thing, and that's kind of where your interest came from starting that. Is there was a need for kind of, I guess, for students to direct their creativity in a way, like giving students tools to be able to use creativity. And so I wanted to, what's your philosophy on maintaining creativity among IGEM students and undergrads while also having them be responsible for the work, like having them consider the safety of synthetic biology, because that's a huge concern for many in the public, and I know IGEM has regulations, so, and we learned a little bit about that, so I was wondering if you can speak about kind of how you approach that.
Yeah, so the creativity side of things, I think providing them with opportunities to engage researchers who have done this for a while and are thinking about questions that are really beyond what you learn in a textbook and what you learn sitting in a lecture, right? So you're learning some stuff that it's new to you, but it may not be that exciting. And then there's people that are doing research that are ahead of that wave already. So they're taking all that fundamental stuff that you learn, and now they're applying it in ways. And the thing is that a lot of undergraduate students don't see that. They don't see where the crest of the wave is. And so providing them with opportunities to go to other universities, for example, and interact with other teams that are themselves working with the very best scientists and people that are doing synthetic biology, and then going as a team, as if we're the Queen's Gales football team and traveling to, another city to play against another team's varsity team. That's what the iGEM competition is. And then there you see these teams that are putting together projects that become companies, that become entrepreneurial things. And so that just, takes the blinders off, and all of a sudden you see all this big science and all these really creative things that come from, again, having people have different inputs to think about problems and to imagine solutions in new ways. So there's that aspect, and then how you contain that in a sense where you have rules, you have guidelines, you have you have sort of a way that you're controlling the type of project that people are doing and making sure that they're sticking to milestones. That's part of the competition, right? So the competition has rules. It has a structure to it. It has standardization, right? We talk about part standardization. All those things are designed to, take big ideas and put them in a form and make the teams work in a form that stays within the lanes. And so that the materials that they produce or the ideas that they produce, they have a translatability. They have a way that you can bank that information that somebody else can use. And I think that's how, again, you have creativity combined with, reining in concepts and reining in activities to make a team work.
Yeah, so many good things there. I think a huge part of synthetic biology that I'm learning now entering iGEM is it really is a community of thinkers, and like there's so much creativity, especially going to the conference and learning about what other people are coming up with and having iGEM meetups and learning what other teams have come up with and working together to build something. And you also talked about standardization, where synthetic biology, in my opinion, from what I've learned, is different from genetic engineering, is that genetic engineering is just a part of synthetic biology, and synthetic biology is really this bigger discipline of applications of engineering. It's standardization, it's simulation, automation. Like it's this entire space that people come together with ideas, and like companies, like you have synthesis companies that sequence DNA, synthesize DNA, and really allow for innovation as a whole. So I think it's so many cool things. And now going back to the creative aspect, you've had the chance to mentor a number of teams, and you've been following iGEM. What's your favorite iGEM project? Like, do you have one? Like, what's the coolest thing you've seen so far?
Yeah, I've liked the projects that are on environmental things the best. I work more in the health side of research, so just for people who are interested, we work on proteins that are involved in mitosis, genome plasticity, and how that is either good or bad. Bad in the sense that certain pathogens use that to their advantage to sneak away from any type of drug we can throw at them. Cancer cells do the same thing. We try to understand that process. We try to figure out how to block it, at least temporarily. But I like to have something else that I do. So, if I have my work topic, and then I have my, softball league or something like that, iGEM is like my softball league. And the thing that I really like is anything to do with environmental stuff. So we've had teams that were trying to do things. One of the favorite teams was modeling ice binding or antifreeze proteins, which, as many of you know, Peter Davies, who's at Queen's University, has been studying ice binding, antifreeze, ice nucleating proteins for decades. He is the person who has really pioneered that field of study. And we had a team that year that was trying to figure out how to use synthetic biology to make antifreeze proteins that could be made and then stored and survive sort of storage conditions where we didn't need to keep things in a refrigerator or freezer until we wanted to use them. We wanted to keep those proteins ultra stable, and so we devised a way to make really simple antifreeze peptides that we used a system that allowed us to circularize the polypeptide. So we know that polypeptides are normally an N and C terminus, amino acids are all through there, and those proteins will fold up into structures that have function. Those proteins are notoriously unstable, but if you can link their two ends together, then they can tolerate extremes of temperature or pH or what have you. And we succeeded in making a class of those proteins as an iGEM team, and then the team published a paper at the end where team members were authors of that publication. And so that was one of the most exciting projects. And then from there, we've done several different environmental things, whether it be trying to detect Borrelia burgdorferi and ticks that you may have encountered when you're walking your dog up in Cataraqui Park, to dealing with water impurity problems at First Nations communities, lots of things like that. Those have been the most fun projects, mainly because, not because of what the team made, but because the team had to immerse themselves in the problem and then devise ways to come up with a solution, educate themselves and their community about the problem and draw attention to it, and then actually learn the science to go about making what they envision making. And so that's been awesome every time. It never lets me down. It's the team. Sometimes the teams do better than others, but every time somebody gains something from it, so it's been great.
Wow. Yeah, I think it's funny that you mentioned that you like the environment side of things and also have your own research background in health. I think similarly, there's so many tools now. Like I know for me, I was also really interested in environment side of things. Just this year, I had to learn how to use some protein prediction tools to try to look at different enzymes that can degrade perchlorates on Mars. I tried to use some prediction to look at the amino acid sequences and then figure out which peptide, like which amino acids were conserved in some of the more efficient enzymes. And I kind of looked at that with no research background, but it's so easy now with with the technology that's available to run things like AlphaFold. There's interfaces that do it for you. Like you don't even have to do the command line anymore. So I was also interested in how have you seen that change over the years? It's been so fast. And with your own work, you also do a lot of protein crystallization things. How has that impacted your own work? And kind of what are you excited about that? Because I know we've chatted a little bit about that before.
It's AlphaFold, AI-based tools, ChatGPT. That's changed everything. So in a matter of a year, 2 years' time, questions that we would be trying to ask and spend years trying to make the materials and make up the experiments or see the things about proteins using experimental tools would take a huge amount of time. AlphaFold has taken that time and cut it down manyfold to see things that we sort of imagined how they might look, and it showed us new things that we never did imagine. It's allowed us to ask much more probing questions and bigger questions and tackle more complex problems. So that's really accelerated research in the same way that, again, AI tools, provide you information right away. If you rather than, let's say you've never coded, you've never used R or something like that to write a code to make some kind of bioinformatics program. Those types of tools just make it so that you don't have to know it exactly, but you know what kind of question you want to ask, you know what information you want. You can have, that tool write you a code in the same way that you can tell AlphaFold, here's the sequence of a protein I want to see the structure of, please give it to me. These programs will write you the code that will do the job. So, maybe it takes away from the virtues that come from struggle and trying to produce something like a structure, or write code, or do this, or write a really good, essay or something like that, it can help you do that right away, so that you can, so your imagination isn't kind of tripped by having to learn a lot of things that just take up time and maybe aren't the most essential part of the problem. So it's like having, a set of assistants around you that each know a bunch of different things. It's the same to me. I use it the same way as, again, if I had a bunch of grad students who all have a speciality, and I can just say to them, can you do this, you do that, you do that, you do that, but I'm just typing that into my computer, and there's the answer. So I think there's some great things about it, and it's really, again, in the last year and a half, it's completely changed how we do things.
Yeah, from everything, like AI. I think AI and the applications of machine learning, like, it's incorporated in just about everything. And it like you said, I think at least right now, it's speeding up what we can do, but it's not yet at the point where you can really creatively think about a problem. It's really good at automating things and those sort of applications. Now, I also wanted to talk a little bit about ethics. That's a huge question I know I always get if I'm talking about genetic engineering or synthetic biology. And so I guess, what is your take on the limitations of, like, what do you think, I guess, the limitations should be from an ethics standpoint?
For using AI tools, you mean?
Well, I think kind of in general, like in gene editing to, like, I think more talking about transitioning more to the engineering DNA side of things. Just kind of your opinion on it, and maybe some things you want people to understand about that. Yeah,
so, I mean, obviously, gene editing in the sense of making things that are incredibly dangerous. So in order to get an advantage over a group of people or a population, or whether or not it's people or we're talking about, something else in nature. So making dangerous things where you haven't carefully looked at the impact, or where somebody doesn't care about the impact, let's say, I'm always going to be against that. So I think anybody, should have some kind of board of people who are in charge of investigating the idea before the idea goes into building some kind of product that can be, whether it's a virus or chemical or bacteria or what have you, or a person who, has different abilities that nature wouldn't have produced in some way over evolution, let's say. Yes, I think those things need to be checked with people who are using an ethical approach. And I mean, you can apply that to AI as well. Anything that, if you get ChatGPT to write you an essay on something that you know nothing about, you read it and you might think, hey, that looks pretty good. That looks better than what I would write, and I think most of that sounds right. But you need to know enough to be skeptical about whether it's right or it's wrong. Whether or not it's right or wrong for you being a university student needing to learn something, or whether the content in that is right or wrong. And that's another ethical type question. It's this being skeptical about what's going to be the impact of this. Is this right? How is it going to be used? And should it be used at all? So I think it's really important.
Yeah.
And we try to teach that in the synthetic biology course for sure.
Absolutely. And I think even just listening from that and what I've seen in the past, I don't think there's anyone else that are advocating more for that responsibility than scientists themselves. Like, even the creators of CRISPR, I've called for a moratorium on the genome editing, side, like germline editing side of things. So I think that's, I think would you agree that you think a lot of researchers have that responsibility, or like in themselves, or?
I think most people do. Most researchers do. I think there are a lot of pressures to bend rules. Whether or not it's a funding question or a competition question. I think There are those issues. There are just knowledge gaps, and those are some of the things that create the problems where maybe it's not a nefarious type of approach that's being applied, or breaking an ethical issue that they're aware of, should be aware of, or maybe nobody's aware of yet. I think that I would like to think that in general, most scientists have an ethical mindset. So I think in general, the thing that drives researchers is that they want the work that they do, the discoveries they make, to stand the test of time. And so if you cut corners and what you wrote a paper about, what you discovered, what you patented, what you tried to translate isn't right, it doesn't continue to work, it was a mistake, that I imagine would really, really hurt. So we do a lot to try to make sure we get it right. So that's why I think research takes so long, at least it does for us, because we test everything. We're people that are, detail oriented, and we like to really understand things, and we do experiments, and we do controls, and we do things like that, and we collect data, and we measure things many, many times, and use statistics and all that stuff that really seems boring maybe as an undergrad, but has a purpose, because again, the smaller the error bars are for the number of replicates that you did, the better the chance that what you're going to tell everybody something means, when you did an experiment, or how something works, the better the chance it's going to be right. You're not going to look stupid.
Yeah.
So, yeah.
Wow. Well, thank you so much, Dr. Alliam. I think that's all for today. There's a lot, we covered a lot there. So.
Yeah. Well, it was my pleasure.
Yeah,
And it was great
so
to have
much.
you as a student in the synthetic biology
Thank you.
course. Lots of great ideas, and I look forward to hearing more about them.
Perfect.